Hi friends, once again welcome back to our next class. Today we are going to learn the topic Oogenesis, that is from the third chapter, Human Reproduction. Oogenesis is the process of formation of mature female gametes. And we have already learned the formation of male gametes, that is the spermatogenesis. Here the process is much different from spermatogenesis of male. The process of oogenesis initiates in the ovary of the female fetus when the fetus is in the womb of its mother. That means when a fetus is formed in the uterus of its mother, this process, oogenesis, initiates. So we can say oogenesis initiates during the embryonic development of the female fetus. And we know in male it starts at puberty, but oogenesis starts before the birth in female child. That is, the female child is about 25 weeks old when it is in the uterus of its mother. Millions of gamete mother cells or the oogonal cells are formed in this stage. All these cells are diploid in nature and it go under mitosis and differentiation. No more new oogonal cells are formed or added after the birth. The cells which enter into the first meiotic division are called primary oocyte. Primary oocyte is also diploid. But the primary oocyte get arrested temporarily at this phase of prophase 1 or meiosis 1. Now, each primary oocyte gets surrounded by a layer of granulosa cell, which is called primary follicle. A large number of these primary follicles degenerates during the phase from birth to puberty, and this phenomena is called follicular atresia. That's about 60,000 to 80,000 primary follicle cells are only left in each ovary. You can see the pictorial representation given here, the development of primary follicle. About the primary oocyte, follicle cells, granulosa cells are added and it becomes primary follicle here. Primary follicle you can see here in this diagrammatic representation. Granulosa cells, this is a primary oocyte, and around the primary oocyte granulosa cells are arranged. A minute thecal layer is also forming here, and now this is called the primary follicle. Primary follicle gets surrounded by more number of new granulosa cells and theca. At this stage, it is called secondary follicle. That means the primary follicles undergo further development to become secondary follicle. From the secondary follicles undergo the transformation to form the tertiary follicle. During the development, the formation of tertiary follicle, you can see that more number of granulosa cells are added here. This is the secondary follicle. You can see the fully grown oocyte is here at the center. Around that the granulosa cells are added. More granulosa cells are added and a presumptive theca layer is also forming here properly. Okay. More granulosa cells and the theca layer is organized into outer theca externa and inner theca interna. A fluid-filled cavity develops within the cell, which is called the antrum. You can see a fluid-filled cavity is developing here, and that's known as the antrum. This is the characteristic of the tertiary stage. So we can say that the thecal layer is there, more granulosa cells are there, and a cavity, fluid-filled cavity antrum is also there around the primary oocyte. Okay, at this stage, the primary oocyte within the tertiary follicle grow in its size and completes the first meiotic division. Remember, at this stage, the early tertiary stage, the first meiotic division completes, and this division is an unequal division where a large haploid secondary oocyte and a tiny polar body is formed. Both the secondary oocyte and the polar body are in haploid condition. Okay. After the birth, childhood, up to puberty, first meiotic division continues prior to ovulation. Now you can see the pictorial representation, that is the maturing follicle cell, the tertiary follicle cell. The thecal layers are developed there. The primary oocyte is also, we can see here, the cavity antrum. It undergo the first meiotic division, reductional division. A polar body is forming and the secondary oocyte is forming. Okay, this maturing follicle, that is the advanced stage of the tertiary follicle. The theca interna and theca externa are well developed. This is the future corona radiata that is around the secondary oocyte. Why? Because the primary oocyte completes its first meiotic division and this is the secondary oocyte. You can see the secondary oocyte large cell and the first polar body inside it. First polar body there inside it. Again the development continues. The thecal layers develop well. The antrum cavity enlarges. The antrum cavity enlarges. The thecal layer develops well, and the tertiary follicle undergo further changes and it becomes a mature Graafian follicle. So this is a mature Graafian follicle. You can see the various parts of it. The theca externa, theca interna, the basement membrane, the member of membrana granulosa, that is the follicular cells here, and the cavity antrum, which is filled with a fluid. Okay, granulosa cell, secondary oocyte. Around the secondary oocyte also some changes are occurring. At this stage you can see a new membrane layer, that is the zona pellucida, is forming here. Zona pellucida is forming here. Okay, and the Graafian follicle rupture at this stage and releases the secondary oocyte ovum from the ovary. This process is called ovulation. It is a hormone controlled process. So the secondary oocyte starts meiosis division 2. It continues, the division is continuing, but it proceeds up to metaphase stage and it temporarily arrested there. Further divisions are starting only after the arrival of the sperm. the sperm need to reach and fuse and to induce the further division or the completion of meiosis II. The completion of meiosis II results in the formation of functional egg or the ovum and a second polar body. You can see the mature Graafian follicle here. This Graafian follicle with theca interna, theca externa, the future corona radiator. You can see the first polar body inside the secondary oocyte, and this follicular layer is rupturing and the secondary oocyte is released from here. So this process is called the ovulation. So we can see within the secondary oocyte the second polar body and the associated part, the zona pellucida and corona radiata is formed around the zona pellucida. The next stage what is happening is the mature oocyte or the ovum undergo the completion of meiosis II, the division II, and it is also an unequal division that results in the ovum and the second polar body. The second polar body also divides meiotically, and as a result we can see that 3 polar bodies and a mature ovum is forming at the end. Now let us see the schematic representation of the oogenesis. Have a look into it. The oogonial cells that are forming from the primordial cells, the oogonium, both are diploid 44 plus XX. Here the female development of female ovum, XX belongs to female. Okay, let's come back. The oogonium with 44, 45, 46 diploid condition, and it undergo further development, and which is going to enter the first meiotic division is called the primary oocyte. Primary oocyte is also diploid 44, 45, 46. The first meiotic division is taking place. It's an unequal division. We know that a large secondary oocyte 22x, that means 22, 23, and first polar body, a tiny first polar body, this is also 23 chromosome number. And during the second meiotic division, that is the secondary oocyte divides unequally to form a mature oocyte and second polar body. Both the mature oocyte as well as the polar body, both are haploid in nature. Meanwhile, the first polar body also, along with this, undergoes second meiotic division, results in 2 polar bodies. So total, at the end of oogenesis, a mature oocyte, functional ovum, and 3 polar bodies are forming, 1, 2, 3, 4. But we have seen that in case of male, the spermatogenesis, which results in same sized functional 4 haploid spermatozoa. Here only 1 is functional, whereas other 3 are polar bodies which will degenerate afterwards. The oogenesis, once again another flowchart you can try to understand. The primordial germ cells enter into the embryo and it undergo differentiation, and the oogonium is formed, which is diploid. This again undergo further mitotic divisions and increases in large number. And at this stage, along with that follicular development, everything is taking place parallelly. You know that primary follicle, secondary follicle, tertiary follicle, mature Graafian follicle, etc. are also forming along with that, but here only the development of oocyte explained. Okay, mitotic division, and it undergo first meiotic division. The cell which entering into the first meiotic division is called the primary oocyte. And first meiotic division, this is the onset of first meiotic division. This is the first meiotic division that results in unequal division, results in a large secondary oocyte and the first polar body. Then it undergoes second meiotic division and ovum and second polar body is forming. That's about the oogenesis. Here is a diagrammatic sectional view of ovary shown. Let's have a look into it. We can see that the developments, we can also the blood vessels and the primary follicle cells are here. In the primary follicle cells, the primary oocyte, around that the granulosa cells, granules are added, granulosa cells are added, and it's developed into primary follicle. It forms the secondary follicle and initiation of tertiary follicle where the antrum cavity is developing, and mature tertiary follicle is showing here, tertiary follicle with antrum. Okay, then it again undergo further development to form the mature Graafian follicle. So this is the secondary oocyte, antrum cavity, theca interna, externa, granulosa cells, everything is there, and here it ruptures and the ovum is released, and that process is called ovulation. So this released ovum enter into the fallopian tube and it moves through the oviduct and finally implanting into the uterine wall. Then what happens to the follicle cells? The follicle cells which surrounds this ovum, it transforms into corpus luteum, which synthesis and secrete hormone progesterone, which is necessary for implantation and further development of the embryo. In the absence of fertilization, the corpus luteum is degenerating. So this pictorial representation is important for the theory as well as in the practicals. Let us have a look into another schematic representation where the follicle cells, how it is developing along with the ovum. This is the germ cell cyst, and from there the primordial germ cells are forming, and it forms the primary follicle. Primary follicles, some granulosa cells are added over the primary follicles. Along the primary oocyte, some granulosa cells and formation of thecal layer is started, and it develops to the secondary follicle. More granulosa cells are added and the thecal layer is further developing into theca interna and externa. And in case of the secondary follicle, further it develops initiation of the tertiary follicle level. When it enters to the tertiary follicle level, theca interna, theca externa, etc. are well developed. Then a cavity is developing which is called antrum. That is why this term, this stage is represented here, antral. And at the same time, at this time we know the primary oocyte undergo first meiotic division and a tiny polar body and a large secondary oocyte is remaining within it. Next, prior to the pre-ovulatory, that is Before the mature Graafian follicle further developments, the antrum cavity develops well. Theca interna and theca externa are forming. At the same time, here around the secondary oocyte, a new layer, that is the zona pellucida, and later the corona radiata, etc. are forming. And next, the mature Graafian follicles undergo ovulation, rupturing of these follicle cells, and the secondary oocyte is releasing. And this may enter for fertilization, and the follicle cells develop into corpus luteum, which is needed or necessary for the synthesis of hormone progesterone. Now let us see the hormonal control of oogenesis. How oogenesis is controlled. You can have a look into the schematic representation of the hormonal control of oogenesis. The hypothalamus is secreting gonadotropin releasing hormone, which stimulates the adenohypophysis anterior pituitary, and it secretes the hormone FSH and LH. Both FSH and LH stimulate the ovary, and it produces estrogen. Okay, FSH helps in the production of estrogen, and it undergoes some changes in the endometrium of the uterus for the development, also in the ovary. The stimulation, follicle stimulating hormone helps in the development of follicles, that is in the ovary, and ovulation is taking place. After ovulation, the corpus luteum is forming and the progesterone hormone is formed. So presence of progesterone hormone helps in the uterus for implantation, and it also gives a negative feedback to the pituitary and hypothalamus, which inhibit the further secretion of GnRH, FSH and LH. Now let's discuss some of the differences between the spermatogenesis and oogenesis. A pictorial schematic representation is given here. First we have a look into it. The primary oocyte in case of female and male, that is in oogenesis and spermatogenesis, both are diploid. So it is dividing unequally to form one polar body and one large size secondary oocyte, but in case of males it is same sized spermatocyte. Okay, and this primary oocyte is dividing into primary oocyte divides unequally to form a tiny polar body and large secondary oocyte, but in case of male it is same sized secondary spermatocyte. And in second meiotic division, it produces the ovum, and the first polar body divides equally to form next polar body. So total at the end 3 polar bodies and a functional ovum is formed. Whereas in case of male, we can see here the secondary spermatocytes divides equally to form 4 equal sized spermatids. The spermatids undergo spermiogenesis, and here only one is shown, but you know that it is 1, 2, 3, 4 spermatozoa or sperms are formed at the end. Now let's see the comparison. That's about today's class. I hope that oogenesis is clear to you. Very soon we'll see in the next session. Thank you.